Hi, all. Well, we're back for another week, and hopefully, we'll get some of our video issues straightened out. And today, first thing I want to do is to get you your first set of problem sets. So, this will be the first of the problem sets. I know we're running a little bit late on these, but we're going to get started. And so, as a result, we're going to talk a little bit about a couple of different ideas that we need to discuss to get to the point where we can do the problem sets. So notice I'm sharing a couple of these problems. There is a handout that has a list of all of of the problems we'll discuss here, but also some we will not discuss and that you'll answer. There is a Word document. Please fill out the Word document, and with the answers, you can do it by computer because it's a Word document, and then just upload it to Blackboard. Or if you want to do it by hand, you can fill in all the answers by hand and then just submit pictures or scans into Blackboard. Either way works for me. I want to try to make this as painless as possible. So notably, in genetics, we talk all the time about numbers. We're constantly counting, as you know, from counting corn kernels. We'll be doing in lab this week. I'll be giving you some data from Drosophila crosses that we've done. And here you're going to see, we're going to talk a little bit about statistics. We've already done chi-square statistic in class. And if you think it's boring, you've got the wrong numbers. Certainly, those of you who are following the information about the COVID-19 infections during the pandemic and looking at the increase in frequency that we're currently going through now, there are a lot of emphasis on the statistics. How many people are infected? How many people are tested? How many people are getting sick to the point of hospitalization? Terribly, how many people are dying? And how do you evaluate those numbers? Well, this is the beginning of it all. I should note that one of the questions that came up during the chi-square analysis is how to analyze and to think about probabilities. So I thought a good place to start today was to actually look at something you're fairly familiar with, something you probably called in the past the bell curve, but is actually called the normal distribution. And the normal distribution is a situation where numbers of individuals or the frequency of individuals in any continuous measurement, when placed, the x-axis would be the measurement itself, and the y-axis would be number of individuals or perhaps the frequency of individuals. In the normal distribution, the graph of distributions is actually symmetrical around the mean. So you can see from this that the mean is in the center line, and there's always plus or minus what we call standard deviations on either side of the mean. And one of the characteristics of the normal distribution is that there is a standard percentage of individuals that could be accounted for underneath the curve at both plus or minus 1 standard deviation, plus or minus 2, and plus or minus 3 standard deviations. And you can see from the horizontal lines above the graph, there are 68, 95, or 99% of individuals accounted for that underlie the distribution, depending upon how far away from the mean the measurements are. So you've done this in Bio 1, probably Bio 2. You perhaps have had a statistics course. The critical element here is that if we had two separate distributions, so let's say we wanted to look at height between men and women, we would have two separate distributions. And how would we know if we wanted to compare the two distributions and wanted to know if the means were different because of chance or because of some underlying mechanism. Well, we would plot the two different distributions, and basically we would say if the means of the two different distributions are more than 3 standard deviations apart, we would say that there is a 99% chance that these distributions are not different due to random chance. That is, there is an underlying difference. Of course, very rarely do we get anything that is 3 standard deviations difference between the means. In general, we tend to think 2 standard deviations of the means because that's 95% of the individuals. And if you think about it for just a moment, that's where this idea of you have to have a p-value or a probability of 5%, that is 5% chance that the differences between the means are due to random processes and not an underlying mechanism. And so this is exactly where we get that idea from. Now, notably, take a peek at the typical chi-square statistic. Notice that the chi-square statistic is not symmetrical around the mean, but rather is distributed from a maximum value of 0, which would be 100%, all the way down to asymptotic to 0. And of course, it doesn't ever really get to 0, but it's asymptotic. And usually we see that there's a 5% distribution. In this case, this is the degrees of freedom of 1 here, and this is a degrees of freedom of 4 here, and we have a critical value of 7.8, which means the p-value would be 5%. And again, that means if you have a critical value of greater than 7.8, there is only a 5% chance that the statistic is due to random chance. So that means we're seeing a significant difference from our expectation. And I think that was a little bit of a problem with some of our interpretations of the chi-square statistic. So hope that helps a little bit. I hope the visuals make a little bit more sense and you can relate it to the ideas underlying the normal curve. Now let's talk about our problem sets. I thought, since we're going to be doing quite a few Punnett squares in this problem set, I thought what I would do is show you the original Punnett squared. This is actually a scan of one of the drawings of Reginald Punnett from 1908, showing his idea. So this is the original Punnett square. Not quite what you probably envision from high school biology or even Bio 1. So notice that at the top, we've got a black and filled in circle and a white circle, and those are annotated as being P1. Those are the parental lines or the parents. Notice that the gametes of the parents, the black filled in circle has both black gametes and the white filled in circle has both white gametes. This indicates that those parents are pure line, that is, they are homozygous for that particular trait. And of course, notice that part of what Punnett did was to talk about the ways in which the gametes can combine. And we're going to find out this idea of gametic combinations is extremely important in genetics, from here to doing Punnett squares, all the way to when we talk about population genetics and talk about the rules of evolution. So, of course, if both individuals are producing only one type of gamete, there is only one type of F1 individual, that is the F1 or the first filial cross. is heterozygous for black and white, and you can see the individual from the F1 population indicated by a semicircle of black and a semicircle of white. And then here comes the fun part. Here's where the square comes in. Recognizing that each individual in the F1 population can make two types of gametes, either black or white, in a 50-50 combination, resulting in 4 different types of F2 zygotes. Notice one is black, one is homozygous white, and the other 2 are heterozygous black and white, but Punnett distinguished between whether or not the black came from the paternal or the maternal origin. So he really did not indicate the 1:2:1 genotypic ratio that we're familiar with. He really had a 1:1:1 ratio because he was thinking about the parental origin of the gamete. Of course, we have kind of a different view of this today. And what I would like you to do now is look at what really is the most important problem we are going to do all semester. And the reason I say that is this is a classic question in genetics. We are going to return to it over and over again. And when we talk about genetic counseling, you're going to find out why this idea is so important. And it's really a very simple question. We're talking about black coat hair or pelage of guinea pigs. Black is dominant to white. White, being recessive, is only expressed with the homozygous small b genotype. And notice the way that the question is phrased. When a pure line black guinea pig is crossed to a white guinea pig, what fraction of the black F2 is expected to be heterozygous? Now think about it for a minute. First of all you're given is the original parents. Nowhere in the question does it even mention the F1. So genetics questions very often will give you the parents, and it is assumed that you know how to get the F1 population so that you can answer part 2, which is what fraction of the black F2 is expected to be heterozygous. So you know that the black guinea pig is homozygous, you know that the white guinea pig is homozygous recessive, and so the F1, of course, just as Punnett indicated in the previous slide, the F1s are heterozygous, and in order to get the second filial generation, you cross 2 heterozygous individuals, and just as Punnett's expectation, we have particular expectations for our phenotypic and genotypic ratios. Now, I want you to stop for a minute, just pause the video, and just do a quick Punnett square and answer the question. What fraction of the black F2 is expected to be heterozygous? Pause. Hopefully it came back. The key here is in reading the question. A lot of people look at this and they say, oh, it's 50%. But because you have 2 heterozygous individuals. However, let's reread the question. It says, what fraction of the black F2 is expected to be heterozygous? So we only have 3 black individuals and 1 white. Remember our phenotypic ratios, 3 black to 1 white, which means of the 3 black, what fraction are heterozygous? And of course, we know our genotypic ratio is 1 homozygous cap B, 2 heterozygotes, and 1 homozygous small b, which means that 2/3 of the black F2 are heterozygous. So this is a very different kind of question. And hopefully some of you got it right. Some of you may have gotten it wrong. Don't worry about it. It takes time to get used to the way genetics questions are asked, okay? Next up in this question is, if a black female guinea pig is test crossed. Test crossing is another one of those terms that you're assumed to know the definition. Anytime something is test crossed, an unknown is crossed with the homozygous recessive progeny. So if I say a black female guinea pig is test crossed, what I'm really saying is we have a black guinea pig. Well, what's its genotype? And the answer is we really don't know. It could be homozygous cap B, or it could be heterozygous cap B small b. So we don't know. We want to find out what it is. And the easiest way to find out is to cross it with a homozygous recessive, in this case, homozygous small b. And in this case, we produce 2 offspring in each of 3 litters. That means there are 6 total offspring, all of which are black. Again, I want you to answer the question, and this will be on the Word document that is in Blackboard. What is her probable genotype? What do you think? Here is the key question in genetics. Not just what do you think, but how confident are you that the expected genotype is correct? And not just very confident, kind of confident, maybe confident, not so confident, not confident at all. No, none of that. The question is, what is the probability that the expected genotype is true? So I want you to think about this a little bit, and how are you going to answer that question? So this is the crux of the entire course, is to be able to answer these 2 questions of really a very, very simple genetics problem. So let's take a look at another phenotype. This is one of my favorites. Obviously, I like chickens. We have a situation here. We have a variety of different types of chickens. Here is the classic single comb. This is something called a rose comb. See how bumpy it is? It also has this little gene here called spike. Oops. There we go, we're back. There is something that is called a pea comb, which looks like you're looking at a pea down the center of the forehead as if it were split open. There is the cushion comb, sometimes called a pin cushion, but usually called walnut. Then there is this is really an interesting, this is a sultan head. It has a tiny little comb and has this font or fountain of feathers coming off the top of its head. They're really quite beautiful. There's the buttercup, and then the strawberry comb. So we have these very, very different comb types, and in every case, we have multiple genes that influence the type of combs that a chicken has. Here are some real pictures of what they look like. Here is the single comb. Here's the pea comb. This is a beautiful picture of a rose comb chicken. This is actually one of mine from Penn State. Somehow we keep changing slides. And here are 2 different individuals with walnut combs. And you can see why sometimes it's called a pin cushion. It looks like a pin cushion sewers might use. So these are the different types of combs. And what are the underlying genes for these different combs? Well, we really have 2 different loci here. There is the pea locus and the rose locus. And the pea and the rose, depending upon their orientation, result in all 4 of these phenotypes. So let's take a peek at what we might see if we take an individual that is heterozygous at both loci and what kind of a phenotype we expect to see. Now, notably, P is dominant to single, Rose is dominant to single, but they are two different loci. So in order to have a single comb chicken, we need to be homozygous recessive at both the P locus and the Rose locus. In order to have a P comb chicken, we must have a dominant P and a homozygous recessive R. In order to have a rose comb chicken, we need to have a homozygous recessive P and a dominant R. And a walnut or pincushion type chicken has to have both a dominant P and a dominant R. So you can see in a situation here, first thing you have to do is make your gametes. So here we've taken an individual that is heterozygous at both loci, that is cap P, cap R, small r. When that individual is capable of making 4 different types of gametes, cap P, cap R, cap P, small p, cap R, small p, small r. And of course, our phenotypic ratio becomes, and count up the reds, these are all our walnut comb individuals, okay, 9. Here are our P comb in green, 3. Our rose comb in purple, 3. And our single comb, 1. So we've got our 9 to 3 to 3 to 1 ratio. Please remember that when you answer any question regarding ratios, you must include a label. You probably learned this in chemistry. You probably recall it from high school, but you must say 9 walnut to 3 P to 3 rose to 1 single. So you have to be very, very clear about which of the phenotypic categories you are defining when you give a ratio. You must, on these problem sets and on the exams, you must include your labels or it's incorrect. One of the things that's interesting to do is to see what happens to the genotypic ratios. Now, genotypically, it's very easy because if all you have is a mono hybrid F2 cross, we already talked about you have a 1 dominant homozygous, 2 heterozygous to 1 recessive homozygous individual, so a 1 to 2 to 1 ratio. Well, I color-coded this the same Punnett square as in the previous slide to show you that what you end up with is a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio. So you can see, you're beginning to see 1 to 2 to 1, 2 to 4 to 2, 1 to 2 to 1. And once you begin to recognize this pattern, you should be able to extrapolate to multiple genes. So I want you to think about fair game for an exam is what would be the phenotypic and genotypic ratios in a tri-hybrid F2 population. Think about that one. As we begin to increase the number of genes that we look at, it's a good thing to take a peek at blood typing. Blood typing you should have been exposed to, certainly in high school biology. We don't do too much of it in Bio 1 or Bio 2, but this is an important part of human medicine. Notably, the most important blood types, as I'm sure you're aware, are the A, B, and O blood types. And in this case, we have 3 different alleles at the blood type locus. A is dominant to O, B is dominant to O, but A and B are co-dominant. And in fact, we designate the genes with the letter I. So we have I cap A, I cap B, and small I. It's always small I homozygotes that are the O. There's also the Rh type, the Rh phenotype, which has only 2 alleles, cap R and small r. Cap R is dominant to small r. And then, of course, there are 2 other genes. This is the MN phenotype, another marker of self located on the surface of the red blood cells. And these are LM and LN. The L stands for Landauer, who was the person who discovered this blood type. And LM and LN are fully codominant. That is, in order to have the M blood type, you must be homozygous LM. In order to have the N blood type, you must be homozygous LN. And the MN is heterozygous LM-LN. And moving along with these, you have to remember these. These blood types will be on the exam later on. They are necessary. This is necessary information to do the following exercises. And of course, what phenotypes and ratios would you expect among these crosses? And so this was just practice. There are 4 different crosses here, individuals, and be able to look at this individual first and say on the first line, that is an A positive individual, followed by a B negative individual, and what are all the possible outcomes of that cross? This also is in the Word document, so you'll be able to fill these in. And please do all 4. We also see that we're able to reverse the idea. So if we know the phenotypes of the parents, so for example, in number 1 here, we have a B individual, 2 B individuals, and we look at their offspring, and they have 3/4 B progeny and 1/4 O. Well, what does that tell you about their genotype? Well, you should right away begin to recognize this is a 3-to-1 ratio, and the only way to get a 3-to-1 phenotypic ratio is if both of these parents are heterozygous. That is, these individuals are both IB and small I. They are heterozygous, IB and small I. And so you're going to figure out the phenotypes of the following parents as well. Subsequently, we know that by using phenotypic and genotypic information, we can actually look at parentage. And we're going to talk about parentage a lot, especially later on when we talk about DNA fingerprinting and the use of personal genomics to identify parentage. And notice that in the case of disputed parentage, the blood group phenotype of the family is as follows. The mother is AMNRH negative, and the child is BNRH positive. You have to figure out what their genotypes may be, and then try to predict all the possible phenotypes of the father. And so there are a couple of these disputed parentage problems in the problem set. These are kind of fun to figure out. And one of the things that you're going to notice is that any kind of blood testing for identification of parents is only exclusionary. That is, having a blood type that could possibly be a parent does not mean you are the parent, because there are lots of people in the population with that particular blood type. So we're going to talk about that, and we're going to find out that that idea still holds even with contemporary DNA fingerprinting techniques. So this brings us to a time when you guys get a chance to actually do the problems. Please use the Word document. As I mentioned earlier, fill it out. You can do it by typing, you can do it by drawing on the document, or you can print it, fill it out with pen or pencil, and then just submit scans or photographs into the Dropbox. So we're off. I've mentioned the exam a couple times. I know we're supposed to have an exam this past week, okay? But all of the technical details of getting things online have been really put me in a position to have to delay the first exam. My plan is to have it ready on Friday, and it'll be due on Monday. But let's see how the week goes. You never know with all of this internet work. So hope you're all well. Hope you're all masked up when you go outside. Yes, even to the beach. It's a little spooky. And you guys take care of yourselves, and I'll see you shortly.